Hey there, nutritional and functional medicine practitioners. Have you heard of Urolithin A? If so, what are your thoughts on that? Is it the next big thing or is it the next forgotten fad? Let's go ahead and take a look at what the science says. So, Urolithin A clearly comes from at least pomegranates. We'll take a look at that. Is it the next big thing or is it the next forgotten fad? Now, in order to talk about that, we need to talk a little bit about memory lane and the fact that history tends to repeat itself. If you've been around in this industry for any length of period of time, you remember number one, resveratrol. About 10 years ago, man, everybody was talking about resveratrol. Lay people were talking about resveratrol. Magazines had articles about it, a longevity, anti-aging, exercise mimetic type of supplement, calorie restriction mimetic. And it turns out that while cool for sure, the human clinical trials didn't really indicate, it's not as great as people were making it out to be. Of course, there was beta carotene a number of years ago, supposedly anti-cancer, but it seems to increase the risk of cancer, especially in smokers. If you're old enough to remember this, chromium a number of years ago was thought to be the next big fat loss mineral. And not only did it not work, but there's a fairly narrow therapeutic window of chromium, relatively speaking, as well as, if you remember this, depending on the industry you're in, is conjugated linoleic acid, CLA. When this first came out, again, this was the hot new supplement. Everybody was taking it because it was supposedly going to help you lose fat. Didn't work, nor did L-carnitine. DHEA, fountain of youth supplement, necessary hormone for sure, but again, didn't pan out as well. And the list goes on, NAD+. There's a whole bunch of things. When the supplement comes out, and it ends up, quite honestly, it ends up being a little bit disappointing. It's not, you don't need to throw it out entirely, but it's usually not as great as people talk about it to be. So we're going to take a look at urolithin A. So molecularly, here it is. There's a couple of these phenolic rings and hydroxyl groups and a ketone on it. It has low water solubility. It tends to be very stable at physiological sort of influences, so pH and temperature, and that's why it works pretty well in the body. And because of this molecule and certain things that it has, it's supposedly antioxidant, anti-inflammatory, and its biggest mechanism seems to be in stimulating the mitochondria, ATP production, as well as mitophagy, which I'll talk about in a second. So urolithin A, if you don't know, is a metabolic byproduct of certain precursor compounds found in foods. So elagitannins and ellagic acid found in things like pomegranates, berries, certain nuts, oak-aged wine, for example. The body, specifically your gut microbiota, converts those precursors found in foods into a number of different things, metabolites. Specifically, urolithin A is the one that probably has the most studies on it. There's supplements on it, and everybody seems to be talking about it as the next greatest supplement of all time. In terms of its metabolism and bioavailability, I thought this paper really nailed it with this graphic here. So you consume certain foods, again, pomegranates, berries, nuts, and seeds, certain ones have this elagitannins or ellagic acid, goes into your intestines, specifically your gut microbiota. Now, research, as far as I've seen, has identified some specific bugs or bacteria that do this conversion, although I don't think research is entirely clear of everything that takes place in this conversion. However, it's converted. You can see down here, elagitannins, ellagic acid. It's not just urolithin A. There's urolithin A, B, C, D, and an iso version of A here. But then it goes through your splanchnic or hepatic portal system, heads over to the liver into phase 2 conjugation reactions, specifically glucuronides and sulfates. And then it goes into body tissues where it exerts its mitophagy, antioxidant, anti-inflammatory effects, or it can be excreted either in the urine or the poop. That's essentially the role of the journey of these. Now, here are some real general amounts of ellagic acid or ellagitannin amounts in foods. Now, I want to be real clear. It depends on when the thing was grown, where it was grown, when it was harvested, how ripe it was, a number of different factors. You cannot just eat pomegranates, for example, and say, I'm getting X number of milligrams of ellagic acid, because it doesn't quite work that way. However, what you can see here is that pomegranates definitely contain the most that we know of. There's a number of different berries, from raspberries, strawberries, cloudberries contain some, and then nuts and seeds. Walnuts seem to have a little bit more than, say, for example, almonds. And I'm not putting it on here because I don't think chugging a whole bunch of wine is a great way of getting urolithin A. But oak-aged wine supposedly has a little bit of it in it as well. Now, research has also found that there are what are called metabotypes. Some people, it turns out, make a lot of urolithin A in the presence of a certain quantity of ellagic acid. Some people don't turn it into urolithin A, but turn it into one of the other urolithins or different metabolites. And then some people don't really seem to make much at all. Now, I'm going to make a quick comment on this because it's not surprising to see this if certain bacteria are responsible for this conversion. Some people might not have as much of that bacteria, other people might have more. But this is where you really get this divergence of should we be taking urolithin A in the first place or not? Some people say yes, because there's different metabotypes. Unless you get tested, you don't know what metabotype you have. You may not even be getting the benefits of eating pomegranate or drinking pomegranate juice in the first place. On the other hand, I look at this and I say, well, if somebody's gut isn't working well, if their microbiome is dysbiotic, if they have certain infections or something's not going well, and when you look at all the different things that can contribute to a less than optimal microbiome, including specific certain things in the diet, stress, lifestyle factors, antibiotic use, even I'll say environmental antibiotics that people have found in water, there's a variety of things that can cause us to have dysfunctional gut microbiome. And I personally, and I'll say this towards the end, I don't think it's a great idea to just take a supplement because some people can't turn it into urolithin A. Instead, why not focus on the gut? There's a number of different papers showing that we can enhance or improve our gut microbiome quite a bit. And thereby, when we do that, not only might we make more urolithin A from its precursors like ellagic acid, but gut health has far-reaching capabilities for anything. Look up the gut-brain axis. Everybody likes to talk about that, but there's also a gut-kidney axis, a gut-heart axis, a gut-liver axis, a gut-skin axis. So instead of just taking this one isolated supplement or compound, why not focus on improving the gut? But more on that in a second. The primary mechanism of action it seems that urolithin A does is it stimulates this thing called mitophagy. It seems to work on the mitochondria. Mitophagy is essentially this process where if you have an old, decrepit, senile, worn out, kind of needs to be replaced mitochondria, the body has this ability through a number of different pathways, but that pink parkin is one of the big ones, that you get an accumulation or stimulation when the membrane is not working well, accumulation of this pink parkin one, which then sort of activates or recruits this parkin, and you get this ubiquitination, and then the lysosome, it puts like a little flag up sort of, and this lysosome, which likes to digest and break down things, comes floating by in the cytosol, gobbles up part at least of the mitochondria, parts of it or some of it, and can degrade it, recycle it, and put the stuff back into the system to make more new mitochondria. So that's all basically to say that mitophagy is a good thing. If we want to get rid of old, decrepit, dysfunctional mitochondria that don't seem to be working as well, and ideally replace them with some new ones. So that's supposedly the mechanism of action of urolithin A. Now, I personally am not so interested in vitro studies, petri dish studies, or preclinical animal studies. They're interesting to talk about, but in terms of should we be taking a supplement or not, I don't think we need to look at those at all. So we'll look at the human clinical trials, just a handful of them right now, to show supplemental urolithin A, what can we potentially expect in humans? Well, here's a paper that was published in JAMA in 2022, double-blind, placebo-controlled, randomized clinical trial, and they said it was older adults. It depends on what your definition of older is, about 65 to 90 years old. Participants either took a placebo or 1,000 milligrams of a urolithin A supplement a day. I think it was divided into 4 doses. And then they had what were called a couple primary endpoints and a couple secondary endpoints. The primary endpoints was a 6-minute walk, as well as ATP production in a hand skeletal muscle in the hand. And then secondary endpoints was endurance in a hand muscle, as well as a leg muscle, the anterior tibialis, as well as some plasma biomarkers sort of looking at mitochondrial function. Listen, I'm showing the study here, so if you can grab it yourself, you can read through this and you don't need to hear me talk about the whole thing. The 6-minute walk in 4 months improved in both groups, both the placebo and the urolithin A. If memory serves, at 2 months, the urolithin A supplemented group had a slightly better 6-minute walk, but at the end, there was really no difference between the two. And ATP production wasn't statistically significant in either group either. However, muscle endurance contractions did increase a little bit in the urolithin A group, and some of the biomarkers associated with inflammation, like CRP, mitochondrial dysfunction, let's just call it, acylcarnitines, which is a great marker for mitochondrial dysfunction. And ceramides are sort of a fatty molecule that happens largely inside of cells, kind of gums it up a little bit. It's associated with things like insulin resistance, etc. So that was a pretty good study. Here's another paper that looked at sedentary elderly males and females of Caucasian origin, again, sort of in the older generations, depends on what you consider old, 61 to 85 years, with a body mass between 18 to 32, and they demonstrated sedentary behavior. Now, this paper did a couple of things. Number one is it looked at how safe is urolithin A in the first place. And so you can see here, they kind of titrated up the dose over a period of time to see if there's any negative side effects associated with taking urolithin A. And as far as I can tell, in a fairly short period of time of months, there doesn't seem to be any kind of negative side effects. It was a very small cohort, as you can see. In terms of some of their biomarkers, once again, acetylcarnitine, which without getting into the biochemistry is a legitimately decent marker for looking at potential mitochondrial dysfunction, especially when you compare it to serum carnitine as well. And the improved mitochondrial gene expression in muscle. Now, I'm showing you the graphic of what was in this paper as it related to the mitochondrial gene expression, because, I mean, listen, it's an elegant, these studies are elegantly done, I will tell you, that they have the machinery, the methodology, the ability to test these things in the first place, the money, as well as the ability to statistically analyze these. And if you're interested in that kind of thing, this particular paper looked at a few different metrics as it relates to mitochondrial gene expression in skeletal muscle, and by and large, a lot of those improved. The third one I want to briefly show you can look at the title alone. Urolithin A improves muscle strength, exercise performance, and biomarkers of mitochondrial health in randomized trial in middle-aged adults. Now, we took 88 people, middle-aged this time, untrained, overweight individuals who tended to have a low physical endurance based off their initial baseline VO2 max. And then there was three groups: a placebo group, a 500 milligram urolithin A group, and a 1,000 milligram a day urolithin A group for a total of 4 months. Now, there's a whole bunch here. You can pause this. You can take a look at it. They measured a number of things: muscle strength, looking at peak torque in the hamstrings as well as in the knee flexors. And there was a little bit of improvement. Oddly, the 500 milligram group did slightly better in the hamstring peak torque than the 1,000 milligram group. Their VO2 max did improve in the 1,000 milligram group. You can see cycling distance improved. So it seemed like endurance, which relates back to the other study that looked at hand muscle endurance did improve. When you look at the mitochondria, that also makes sense. Once again, certain markers, acetylcarnitines, decreased, specifically in the 500 milligram group. There was some of the inflammatory markers or cytokines also seemed to decrease a little bit. And then there was some additional skeletal muscle findings relating to that mitophagy, Pink-Parkin pathway, dependent pathway, which relates back to the previous study of some of the genetic modifications that might take place in skeletal muscle and the presence of urolithin A. Now, this paper, which I already showed you, I'd have to look back, just triple check, but it didn't have any limitations. Usually a paper will have limitations in it. It will say, here's the paper that we did. In hindsight, here's a few things that might have skewed some of the results, possibly. Maybe it was short duration. Maybe it was a homogeneous or heterogeneous group. Maybe it was, maybe there's a placebo effect or some other control factor that maybe they didn't think about ahead of time, so that researchers can look at that, other researchers can look at that, read the limitations, and if they want to replicate the study, try to remove some of those limitations for the replication of the study to see if it affects the results at all. Now, I say that because I think that there's some ethical limitations on this, although there was a full disclosure on this, and I want to read this to you, and then I'll show you something else. The authors of this particular paper declare the following competing interests. Certain initials are employees, certain initials are board members, and then some more initials of members that were authors of this, of the Scientific Advisory Board for Amazontis, the sponsor of this clinical study. Now, I'm not going to go into specific names because I personally don't know any of these people. I'm just commenting on this on a purely sort of objective view. So if you look at the names in that paper, as well as this one, as well as this one, as well as this one that I just showed you, and one that I didn't show you, but this one talks about the benefits of urolithin A supplementation overcoming the challenges of certain metabotypes of people not actually making enough urolithin A based on the precursors in the diet because of an altered microbiome. All of these papers were essentially sponsored, paid for, or the people authored it, work for, are on the board, employees of a company that sells that supplement. Now, when you look at the amount of human clinical trials using urolithin a large majority of them have some of these same authors, which is therefore sponsored by a company that sells it. Now, a couple quick things I want to point out on this. Number one, that's a huge potential flag and concern. Anybody that has some kind of financial vested interest in the outcomes of certain studies, it has to be concerning. It does. It's just the reality. Now, on the other hand, and I will say they have a ton of money. Studies can cost in the neighborhoods of hundreds of thousands of dollars to even just run one study like this. So they clearly have lots of money. But on the other hand, and to the credit of some of these researchers or there's these papers, they do report occasionally what are called null findings or even negative findings. So and that's to their credit. Usually, if someone has a vested interest, they'll leave out the bad stuff. If they have a negative finding or a negative study, if there's negative side effects, or there was nothing happened, they'll be like, conveniently, they'll leave that part out of the study. They might modify it, not even publish it in the first place. They might statistically modify certain things in the findings, which is peer reviewed. But once again, it has to be a big flag in order for that to be concerned or retracted for some reason. So all I'm saying is the facts. A lot of these people on a lot of the papers done on human clinical trials are paid for in some way, or the paper itself was sponsored by a company that happens to sell this particular supplement. So beyond that, and I can't speak any more to that except for just the facts. Beyond that, so let's talk about something beyond just this molecule of urolithin A. So I already showed you this paper in the slide, but what I wanted to show you once again was the fact that from ellagic acid, urolithin A, which is here, is not the only compound that it makes. There's urolithin B, urolithin C, urolithin D, and iso-A, like I said. And in the case of urolithin B and C, there's not a lot of human clinical trials that I've ever seen on these. There's some preclinical trials or some in vitro studies, but it's not the only thing that comes from it, for sure. And what I want to talk about then is there's thing of the whole food complexity, sometimes called the food matrix, and that is that you can take some of these urolithin A precursor containing foods and look at all the other things that are in there. Now, in many cases, that some are known, some probably haven't even been discovered yet, or those that have been discovered, we don't even know the full story on some of these things. But we have been given these foods over a really long period of time, and they have dozens, if not hundreds, of different compounds in them that have health benefits. And again, you look at these foods and you look at the number of things that there are studies on showing that they're beneficial for. We miss out on all the rest of that stuff, and all we do is we take one particular supplement or molecule. Now, I'm not totally against urolithin A supplementation. I personally probably won't take it, but that's fine. Maybe to my detriment, who knows? I guess we'll find out. But it has some benefits to it. For example, urolithin A, you can standardize and specify the dose. You know that if it's a good company, if you're taking 500 milligrams, you're probably getting 500 milligrams. If you're eating food, that's going to be highly variable, as well as your own microbiome. Concentration is going to be a lot higher than you can get in food. You'd have to eat a lot of pomegranates or pomegranate juice, and then you might have some other problems. And it's super convenient. You just have to basically buy a bottle and pop your pills, and then you're good. On the other hand, whole foods have a whole bunch of other additional benefits that this is not going to have. I'm kind of being kind here when I say limited, but the synergistic effects, there really aren't any. It's a single molecule versus there's potentially a lot of synergistic effects of all the different things that are found in one particular food or food group. The cost is going to be generally higher, clearly, than it is with food, although food's not as convenient. There's limited long-term safety data on this right now that I've seen. It's just months, not necessarily years, especially many years. And in terms of gut microbiome support, it's questionable if this actually does much with the gut microbiome as we've seen right now, although there seems to be some interactions. It's potentially a lot higher when you look at the number of compounds in some of these foods, as well as some of the things like prebiotics. And then other beneficial compounds, again, I'm being kind of kind when I say limited. There's really none except for one, maybe the gelatin cap that it comes in. And it's abundant in these other ones as well. Now, I'm not going to talk about how to improve gut health because the studies are very clear on this. There's a number of different ways. The more dietary diversity that we have, the more consumption of we have traditionally fermented foods, decrease our stress, improve our sleep, or our sleep hygiene as it's sometimes called, regular physical activity, and then avoiding things like alcohol and certain medications that will negatively impact the gut microbiome. This is all really well established in the literature. So then my question is why are we focusing here, taking urolithin A as a supplement, which might improve gut health a little bit, but why not then focus on gut microbiome diversity in the ways that I just talked about? Because when this works well, again, there's a gut-kidney axis, and look it up. There's a gut-brain axis. There's a gut-heart axis, a gut-pancreas, at least a gut, or maybe it's a brain-pancreatic islet axis. But it connects with everything. There's a gut-skin axis. The better our gut works, the better the rest of us work. So why take a single isolated molecule supplement out of a certain food and focus on that when we could be focusing on something else? And if our gut microbiome diversity is good, well, guess what? We get better production of urolithin A as well as the other urolithins, which tends to have improved gut health, which then goes back to gut microbiome diversity. So here's my question, and please, I'm interested in your answer. What's your take on this? Should we be eating healthy foods with dozens, if not hundreds, of different molecules and substances, some known, some unknown, that likely have some kind of synergistic effect that we've been doing for thousands of years at least? or take a single isolated molecule, likely synthetic, I'm guessing, with limited human clinical trials on it, limited human safety data, doesn't have any kind of other synergistic effects to that. I'd love to hear what you think. Go ahead and comment below. And in the meantime, if you like this kind of information, you can always hit like and subscribe to get notifications when we have more videos like this. And if you'd like, you can always head over to metabolicfitnesspro.com, where my wife and I train healthcare practitioners on how to elevate their expertise, deliver life-changing patient transformation, and to help build a thriving practice with evidence-based functional medicine and education. I hope to see you there. Thanks so much.